Thank you, and welcome everybody, and thanks for the opportunity to give a talk. And so I'm new to the graduate program. So for those graduate students that I haven't met, I've only met a few. Please feel free to introduce yourselves to me, and look for emails to try. I'd like to get us all together to go for a happy hour and just discuss the program, so I can learn about it from your perspective. But today is going to be a more of a basic science talk that's going to focus on synthetic and mostly on natural products, and then I'll talk about a synthetic antiviral. Oh, excuse me. We'll be talking about primarily on the anti-inflammatory actions of natural products, and give a large history of our work on the extract known as milk thistle that is formally named Silymarin. And then I'll be talking about this broad-spectrum antiviral Arbidol that was developed in Russia about 30 years ago as an anti-influenza drug that seems to be inhibiting more and more viruses, both in vitro and in animal model systems. And I'll talk briefly about our work on hepatitis C, HBV, and recent data on Ebola virus and Zika virus. So this attractive-looking guy with the hanky on his head would be me. This is in Europe a few years ago. This is Mount Go in Spain, outside of Javia. I was hiking Mount Go and I came across some milk thistle. So this is what the plant looks like. And the extract is derived from the seeds of the milk thistle plant. And so Silymarin. Pardon me? Yeah, and it's actually in Washington State. It's a Class A noxious weed. It's like toxic for ruminants, but it actually Silymarin emanated from European medicinal practices. As I mentioned, Silymarin is an extract from the seeds of the milk thistle plant. Its official name is Silybum marianum L. It's derived from, it was actually appeared in book 4 of the Pedanius Dioscorides, the first doctor who wrote this book on medical matters, or materia medica, referred to as Silubon. And since about the late 60s, when there's a lot of anecdotal data to support its role as a liver protector or make you improve the health of your liver. Since about the late 60s, there's been many studies that really started in Germany showing that this extract prevents liver disease in many experimental animal models. So for example, carbon tetrachloride toxicity, acetaminophen toxicity, ethanol toxicity, amanita poisoning, toxic mushroom poisoning, Silymarin, either through IP delivery or oral gavage, prevents liver disease in these animal models. And it turns out, quite serendipitously for us, it was shown that it's widely consumed by subjects with chronic hepatitis C and HIV infection for its purported hepatoprotective effects. And there's been many animal and clinical studies that indicate that it's well tolerated and safe, and when taken as orally available. It's really in the supplement industry. So we've been probably for the last decade or so really focused on Silymarin. This project started because I saw an opportunity for funding in hepatitis C from the National Center for Complementary and Integrative Health, which is one of the NIH institutes. And they were looking for studies that got at the mechanism of actions of natural products. And so, to cut a long story short, it took us 3 tries and a publication in gastroenterology with Chihiro Morishima that we actually were able to get funding because we showed this extract could inhibit in vitro hepatitis C virus infection. So I'm not going to talk too much about its role in Hep C, but I am going to spend most of my time today on focusing on the anti-inflammatory effects of Silymarin on human cells in culture. And we're focusing mainly on human liver cells, hepatocellular carcinoma cells, as well as primary hepatocytes, T cell lines, and more recently we've expanded into primary human immune cells, including T cells, monocytes, and these specialized type of T cells called mucosal associated invariant T cells. So Jessica Wagner works in my group. We've been together, I think, 12 years, and she generated these data several years ago. So this is a a real-time PCR, RT-PCR expression of a pro-inflammatory chemokine called CXCL10. And human hepatocytes were infected with hepatitis C virus, and Jessica measured CXCL10 at 4, 6, 8, and 12 hours post-infection. And so you can see in the solvent control cells that HCV infection causes a time-dependent increase in CXCL10 mRNA, and silymarin completely blocks that. And in contrast, because we know that silymarin can inhibit HCV infection in vitro, Jessica did this important control where she measured the amount of viral RNA in the cells. And you can see that both the DMSO and silymarin treated cells had equivalent levels of hepatitis C virus RNA, indicating that this is a true suppression of inflammation and not due to a result of suppressing virus replication. Work in collaboration with Martin Prilich and Hannah Miller, who used to be in his lab. We looked at silymarin's ability to inhibit pro-inflammatory cytokine production in primary human monocyte cultures. And so here, there are 6 different cytokines and chemokines, so MIP1 alpha, MIP1 beta, TNF alpha, IL-1 alpha, IL-1 beta, and RANTES, which is another chemokine. And you can see this is a small sampling of 3 PBMC, or sort of monocyte preps from 3 different patients. And you can see that silymarin inhibits both the basal production of these cytokines and chemokines, and it also reduces the induction induced by different TLR stimuli. So here, TLR4 stimulus was used, lipopolysaccharide, to induce, for example, MIP1 alpha, and you could see that silymarin inhibited that. TLR8, which was a single-stranded RNA, induction of pro-inflammatory cytokines, was also inhibited by silymarin. So the previous slide and this slide says that silymarin can block inflammation induced by pathogen-associated molecular patterns. So we also did a little bit more work with Martin Prilich's lab, who is one of the main labs in the world studying these mucosal-associated invariant T cells. And in this experiment, Hannah treated cells with to activated the expression of interferon gamma and granzyme B production from these mate cells with a combination of a cytokine cocktail or a cytokine cocktail with T cell receptor stimulation. And you can see that silymarin, or the cytokine stimulation, induced the production of interferon gamma and granzyme B, and silymarin suppressed that induction. So these data go along with other data that we have in the labs, indicating that silymarin can suppress the basal activity, the basal inflammatory status of immune cells, and cytokine-induced induction of inflammation. So we went a little step further. We got a supplement from CFAR a few years ago, and Erica Lovelace, who really was working in my lab at the time, she spearheaded this effort. And we went into the CFAR repository, the local CFAR repository, and measured immune activation and exhaustion markers in HIV-positive subjects, peripheral blood mononuclear cells. And the reason we did this is because HIV-infected patients aren't dying from AIDS anymore because of highly potent antiretroviral therapies. But it turns out that they have other ailments. They live longer. They don't live as long as the non-HIV infected population. And it turns out that there's a lot of subjects have sort of an ongoing enhanced elevated inflammatory status. So you can see there are these markers that mark T cells, CD4 and CD8 T cells for chronic immune activation, and these markers are CD38 and HLA-DR. The bottom line is that when Erica got these cells and she cultured them ex vivo, and in the absence, in DMSO, the solvent control, and silymarin, there was a statistically significant reduction in these markers of chronic immune activation. And in addition to having these kind of a heightened inflammatory status of their immune cells, people with HIV also, their T cells look anergic and exhausted, and typical markers of those are CTLA4 and PD1. And so Erica also measured the expression of these markers on CD4 and CD8 T cells, and she noticed that silymarin could suppress the inflammation in the context of HIV infection. So this is kind of the sort of the bioactivity that we can survey with natural products like silymarin in the lab on liver cells and human immune cells, because we're interested in inflammation in general. But there's a whole host of other activities that silymarin does to protect cells. So we call this silymarin and cytoprotection. And Jessica Wagner, and a lot of this data here in red are the papers from my lab and the lab of my collaborators. And Jessica Wagner has had an instrumental role in helping us generate all this information. So these silymarin, and with natural product chemists we work with who provide us purified compounds, we've shown that, as I mentioned, that silymarin can block Hep C infection, and we've shown that it can block virus entry by slowing clathrin-mediated endocytosis. And there's also some role for inhibiting the association of one of the viral proteins, NS4B, with lipid droplets. We've also shown that it's anti-inflammatory by blocking NF-kappa B activation. And indeed, many of the cytokines that I showed you are targets of NF-kappa B, which is a transcription factor. There's many effects on many other cellular pathways. We've also showed that it blocks virus-induced oxidative stress. And it blocks, as I showed you earlier, and in some other papers, it blocks T-cell proliferation and pro-inflammatory cytokine production. And a certain form of the extract, a water-soluble version, a derivative of the extract, also can block HIV infection. So the question becomes how can silymarin do all these things? And what we've been particularly interested in is trying to understand how silymarin suppresses inflammation. And the literature is inundated with data on natural products perturbing this pathway or that pathway, or blocking inflammation in this cell type and this cell type, or inhibiting this transcription factor. So we really wanted to take an unbiased view of how silymarin alters immune cells like T cells and hepatocytes. So we turned to systems biology and microarray and metabolomics analysis to get at this question. And Erica spearheaded this effort with a host of collaborators at different institutions. And I wanted to share with you some of the microarray results that Jessica directly participated in. And so here, the results are shown here that at the top, over 4 and 8 and 24 hours, we saw a few genes, less than 100, that were up. There was kind of genes that kind of, if you look at it in these heat maps, you can see that there were these genes that immediately came on at 4 hours, and they kind of stayed on for the duration of the experiment, which was 4 to 24 hours, 4, 8, and 24 hours. On the other hand, there was a set of genes that there was a progressive increase in genes that were downregulated with silymarin treatment. And you can see this in this heat map. In the red are the genes that are downregulated. So there's a few genes that go down at 4 hours, and there are more and more genes that get downregulated over time. So we wanted to know, what are the genes and signaling pathways that are associated with these gene expression patterns? And is there any connection between these genes that are rapidly induced and stay induced and genes that get progressively shut down? Yes?
So if you were to do this sort of experiment with another compound or material that you had those sort of effects, like, I don't have a good sense where this is. Lots and lots of genes, like, how would you compare that to another sort of compound with known effects that you would hypothesize would be similar?
So we didn't do that. That's a great question. Is this just like a standard response of a cell to any kind of treatment? And so we didn't do it in this experiment, but we are doing studies now through collaborators at the Broad Institute and informaticians at Mount Sinai, there's this program called the Library of Integrated Cellular Signatures. So they're basically taking hundreds of cell lines and thousands, like 20,000 compounds, in addition to other perturbations like single gene overexpression and single gene either siRNA or CRISPR knockdowns. And there's all this publicly available data. So we've been able to take this silymarin signature of our significantly regulated genes, and we've been able to input that on on their database, and we get back a list of compounds that are predicted to cause similar or oppositional transcriptional effects. So we're still working on that. We got a supplement at the last year of this R01, and we're also, and what I can tell you is that when we've looked at the individual components of silymarin, there's about 7 or 8 major components. Their transcriptional responses are different, and that's over. This was just a single dose, but now we're doing 6 different doses with this. We have some high throughput access to high throughput screening. So I don't know the answer to your question yet, but it's in progress. So if you look at, this is just a blow up of a section of the genes that were rapidly induced by silymarin. And our bioinformatics collaborators and Erica also figured out some of this, that we actually found that there were several genes associated with cellular stress and nutrient responses. So for example, we saw that the response at 4 hours was indicative of ER stress because we saw transcription factors like ATF4 were activated. And it also mimicked or was predicted to look like nutrient deprivation or suppression of cell growth or anabolism. So these data suggested that silymarin was altering cellular energetics. And when we did some metabolomics profiling with collaborators at PNNL in Eastern Washington, we noticed that glycolytic tricarboxylic acid and amino acid metabolism were all suppressed. And so we're not exactly sure why that happens, but based on this information, we started thinking about other kinases that might be activated. So we turned our attention to adenosine monophosphate kinase. And this is a really cool enzyme in that it really receives signals for the cellular nutrient status. So it gets activated when cellular energy levels or ATP levels are low. And what it does is it inhibits multiple downstream targets, including mTOR, which is the mTOR pathway is a pathway that says, when there's a lot of nutrients, it says build, 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 and grow. But when energy levels are low, AMP kinase shuts down mTOR. And we noticed that silymarin at a couple of doses could activate AMP kinase. So that's the first half. So AMP kinase activation is associated with many of these genes that are upregulated and stay regulated during the time course of this experiment. On the other hand, for the genes that were progressively downregulated by silymarin, Theo Bamler, who's here at the UW, and Erica worked out some bioinformatics analysis. And these data are basically called ingenuity pathway analysis pinwheels. So at the center is the predicted regulator of all the genes on the outside of the wheel or that comprise the spokes. And all of these genes, the majority of in red, indicating that they were downregulated, were the ones that were significantly regulated or inhibited by silymarin treatment. So this shows that silymarin, this later state of progressive downregulation of gene expression is progressive suppression of cellular inflammatory status. So then the question becomes, well, at least my question was, is this suppression of cellular metabolism and cellular energetics linked to silymarin suppression of inflammation? And because we'd shown that AMP kinase is activated by this extract, we asked the question, does silymarin inhibit NF-kappa B via AMP kinase? And the answer is, I could show you more data, but this is kind of, this cuts to the chase. So if you focus on the black bars, this is a NF-kappa B transcriptional activity assay, luciferase reporter gene, in cells treated with, we activate NF-kappa B with TNF, and then we treat cells with silymarin. And you can see in the black bars that silymarin causes a dose-dependent suppression of NF-kappa B, much like we found in the microarray results. And when we used, these are mouse embryo fibroblasts with wild-type AMP kinase, and when cells with a double knockout of AMP kinase, you can see that silymarin did not inhibit NF-kappa B as well when AMP kinase was there. So this suggests that the cell sensing of cellular energy status communicates to the cellular inflammatory status. So, so what do we do next? Where are we going? Is this fundable? I hope so. We're interested in exploring other pathways that lead to this suppression of inflammation. I mean, I kind of related to your question, Noah, many natural products, including silymarin, have been shown to inhibit NF-kappa B. And again, the literature is so confused with this. People say NF-kappa B activity is inhibited, therefore that's the target. And I don't think that's true at all. I think the activity could lie far upstream, and because NF-kappa B, for example, receives so many inputs from different pathways, I still think there's a lot of work to be done to dissect that. We've started in liver disease, and we've moved a bit into chronic inflammatory diseases that HIV positive subjects experience. We'd like to apply these to other inflammatory processes and diseases, so I'd welcome any ideas and suggestions and collaborations with that. And we're starting to, there are some limitations studying silymarin that we can talk offline to conserve time. But we're starting to get into other natural products. And as I mentioned, this library of integrated network-based cellular signatures was this LINKS program. We're in the process of testing all of the components of silymarin, some of their metabolites, and other natural products. They have this high throughput custom microarray profile, microarray system that they have 1,000 genes. And that's all they profile is 1,000 genes. But when you profile these 1,000 genes, the transcriptional response of these 1,000 genes captures the response of 80% of the human transcriptome. So it's a very economical and high throughput way to figure out what's happening. We're also starting to get into some work with fungal metabolites. So we have honored to have student Fletcher, our rotation student, MLS rotation student, Fletcher Hill, who's here today. So I'm trying to give him a perspective of where his project fits in the grand scheme of things. And the bigger question, which we're planning on submitting a PO1 to the NCCIH, is how do combinations of natural products achieve biological synergy? There's a growing area, emerging field of network pharmacology that says that drugs that hit multiple targets are more effective than the one drug, one target hypothesis. And that actually aligns really well with what complementary and alternative health practitioners view in traditional Chinese medicine, for example. It's the combination of the components in an extract or multiple extracts that gives you bioactivity. So we're developing some hypotheses to fit into 3 projects and working with natural product chemists who are really good at defining the chemical complexity and the chemical signatures that suppress inflammation. And we're going to be involved in this second project where we're looking at using this sort of high throughput technology of affordable microarray processing. We're looking at network perturbations that underlie botanical natural product suppression of inflammation. And then we have some informaticians who we've enrolled who are going to get at data mining mechanisms of actions of botanical medicines, again, combining all the power of this LINKS program. So that's this natural stuff. Now we're going to be synthetic. And I want to tell you about this really cool molecule that has really been, for about the last 10 or 11 years, it's kind of been a hobby of the lab. And now we're trying to get serious and get funding. And this compound is known as Arbidol. It's its full name here, PubMed compound ID. It's also known as umifenavir. And this was developed in Russia about 30 years ago or more as an over-the-counter anti-influenza virus drug. And it's been used for 30 years over the counter. I have capsules in my office when I was over in Russia. And there are many clinical studies, more and more being published in Western journals, showing that this compound can inhibit multiple seasonal flu isolates in vitro in animal models and reduce severity of disease in animal models. And there are some clinical studies that are indicating that when you take Arbidol, as soon as you feel like you're getting sick, it reduces the duration of hospital stays and severity of symptoms. So it's really popular in Russia. It's very popular in China. And so about 10 years ago, over 10 years ago, we had a visiting scientist in the lab from Moscow, and he wanted to learn. We just gotten the HCV culture system in the lab, and so he brought Arbidol with him and wanted to test it. And basically Yuri Boriskin was able to show that here's a Western blot of HCV infected cells that were pre-treated for 2 days, 1 day before, at the same time of infection, or we delayed the addition up to 2 days post-infection. You can see that Arbidol, when you pre-treat cells, gives pretty significant suppression of hepatitis C virus infection, and that's true at the protein level and at the RNA level. And if you look at just infectious virus foci, you can see that this pre-treatment really suppresses hepatitis C virus infection. So it's been shown that Arbidol can inhibit influenza virus entry, and I'll show you some recent data from other labs about how that happens in a minute. So with a collaborator in France, Yves Pecheur, with whom we've been working with for over 10 years now, she tested Arbidol in this fusion assay. And what this assay is a surrogate for, it takes cell culture derived viruses of hepatitis C virus or viral pseudoparticles, and it mixes them with synthetic liposomes that are fluorescently labeled, and they're so heavily labeled that there is no fluorescence. So when if these two structures mix and engage, you actually see this recovery of fluorescence over time. And so this is a surrogate for a point in the hepatitis C virus entry pathway into cells. So Yves was able to show that Arbidol, here's the fluorescence recovery with a couple different genotyped pseudoviruses. You get nice recovery of fluorescence, but Arbidol causes dose-dependent inhibition of this recovery of fluorescence. And this indicates that Arbidol is inhibiting the fusion of HCV pseudoparticles with membranes. So since that time, there's been a growing list of viruses that Arbidol seems to be able to inhibit both in vitro and in vivo. So there's the original papers on influenza, RSV is inhibited, polio viruses are inhibited, chikungunya virus is inhibited, and we've shown Hep C is inhibited, and hepatitis B virus has also been shown to be inhibited. So a few years ago, I took advantage of the NIAID in vitro antiviral screening program, and I convinced them to test Arbidol against all their viruses, and they pretty much do every virus. And the cool thing is, it's free. You just have to write a small proposal. So, and at the same time, about 2 years ago now, the Ebola virus outbreak was happening, and so I managed to convince Tom Geisbert at UTMB in Galveston to test Arbidol. And so about a year ago, we put all these data together, and we showed that so we added some new viruses to the list that are inhibited by Arbidol. And we showed that Ebola virus Zaire, human herpes virus type 8 or Kaposi sarcoma virus, and Tachyrebia rhinovirus was inhibited by Arbidol. And we also confirmed the prior studies that showed that hepatitis B virus and poliovirus type 3 were inhibited by Ebola virus. Sorry, by Arbidol, confusing my viruses with drugs. This is a slide showing that Arbidol inhibits an early stage of Ebola virus infection. And so similar to the work that we did with Hep C, we pre-treated cells for 24 hours with drug, or we added drug at the same time as virus, or we delayed the addition of drug to 1 hour and 24 hours after infection. And you can see that Arbidol, when given 24 hours before infection, caused multi-log suppression of virus infection, and this was dose dependent. If you waited till 24 hours after infection to add Arbidol, it didn't really have much effect, and the effect waned also as you, the closer and closer that you got to the point of virus infection. So these data suggest that Arbidol is blocking Ebola virus entry, and we've tried twice now to submit an R21, but we can't get any traction on this. So we're rethinking that. And then the last thing I wanted to show you, I'm jumping on the virus du jour. I tried Ebola, now I'm trying Zika. So we have 6 different Ebola, sorry, confusing my viruses again. We have Zika isolates in the lab. We have the original Uganda isolate, which is in the African lineage, and we have several Asian lineages, so the Puerto Rico isolate, a couple from Panama and Mexico. And from Mike Gale, we got a Brazilian isolate that was circulating recently in Fortaleza, a northern Brazilian city. And you can see this is a Western blot showing the Zika virus envelope protein and the Zika NS5 protein, and that Arbidol is appears to be dose-dependently inhibiting Zika virus in these assays of these different multiple isolates. So I've got an R21 out there. We submitted that in October, trying to get some money to follow this up. We're also working with some local collaborators at who have an R21 studying sexual transmission of Zika. Interestingly noted, when Zika kills cells, so it's a lytic virus. So here's an example of uninfected cells or mock-infected cells, and when you infect cells with Zika, you see fewer cells. You see cells rounding and floating in the culture, and we noted that Arbidol could completely protect against Zika virus-induced cell death. So we're interested in pursuing, we think it's blocking virus entry, so we've proposed those types of studies. But, no, that's just the, yeah. Well, it could certainly lead to certain forms of transmission of Zika virus, right? No, this is the solvent control, but that's a good point. Thanks, Brad. Appreciate that. So the question is how does this drug, if it's inhibiting hepatitis C virus entry, how does it inhibit, we think it inhibits Ebola virus entry, and I think it probably inhibits Zika virus entry into cells. So how can, these are very different viruses, how the heck can this one drug hit all these different viruses? And it also, if you notice on the list, if you know your virology, it inhibits both enveloped and non-enveloped viruses. So there's the question arises is whether Arbidol is targeting the virus or targeting the cell. In my mind, a broad spectrum antiviral is most likely going to target the cell, which all viruses need to grow in. That's been my hypothesis for many years until recently. So these are data from Irina Leneva. She was on the original team that developed Arbidol and is still active as a researcher in Moscow. And working with a group in the UK, they were able to select for, in cell culture, viruses that were resistant, influenza viruses that were resistant to Arbidol. And they mapped the position of the mutations to the hemagglutinin protein, which is interesting because there are really no hemagglutinin inhibitors. All the known inhibitors of influenza virus target neuraminidase, like Tamiflu, also Tamiflu, for example. And they suggested that where arbidol was, these resistant mutations allowed it to stabilize the conformation of hemagglutinin, and it prevents the fusion steps. Hemagglutinin needs to rearrange and go through a massive conformational change for the viral membrane to fuse with the cellular membrane. And so they thought that that's what was happening. And I wasn't really so sure about this because it's been, a few years ago that this was published. But just last month, Ian Wilson, who's at Scripps, he published the crystal structure of arbidol docked to the hemagglutinin protein, and it interacts in 3 places, and they really showed convincingly. I'm not a structural biologist, but you can see that's pretty nice binding, and they described all the hydrogen forces and the pi stacking interactions. And he really showed that arbidol acts as a molecular glue, and it stabilizes this prefusion conformation of hemagglutinin, and it really inhibits this massive conformational rearrangement that happens when the viral membrane fuses in the low pH environment of the endosome. So I'm starting to, rethink that there could be, viral targets for this drug. There's also one paper in chikungunya virus that they also selected drug-resistant mutants that also map to one of the viral envelope glycoproteins. But my favorite has been, because this is broad spectrum, it's that arbidol targets the cell. And there was a recent really cool paper that came out that showed they compared the regular arbidol with the positively charged or protonated form of arbidol, and they found that that both types of the molecules rapidly integrated into cellular membranes, and that the protonated form engages in parallel with the membrane, but the non-protonated form kind of goes in perpendicularly into membranes. And they showed some other data that suggested that the protonated form of the drug could more strongly interact with negatively charged phospholipids. So that's one argument, that it's targeting the cell, and other investigators, including Yves Pechura, showed that this drug can really engage cellular membranes. The other question is whether arbidol targets a cell, and this is some movies that Yves Pechura and Julie Blazing, who was a grad student in her lab, we collaborated a few years ago. So in red are fluorescently labeled viruses, and in green is a clathrin molecules expressing GFP. So this is spinning disk confocal microscopy, and it's hard to really follow. But if you watch, you can see the red spheres move around, and Julie was able to measure or track the movement of the viral particle into the cell. And so what she found in untreated cells, that clathrin co-localization lasts for about 60 seconds, and there's high viral speed, and the virus moves from the outside of the cell into the inside of the cell. And in contrast, oops, sorry, I wanted to show that movie again. How do I do that? There we go. In cells treated with arbidol, it's like the virus is stuck and becomes, it's blocked in clathrin-coated vesicles. They also backed up these studies with other imaging with markers of early endosome, so you could measure how long it took for the virus to get into an early endosome. And in arbidol-treated cells, it never seemed to make it there. So these and other data that Yves and Julie have generated indicates that arbidol seems to slow clathrin-mediated endocytosis. So what's next? The F word. I've been saying that a lot because it's been really hard to get funding for this, but we remain hopeful. As I mentioned, this R21 with Zika is pending. The Ebola has been unsuccessful despite 2 attempts. And I find it hard to reconcile that, because this drug is used clinically. So, why wouldn't you want to fund this? Give me the money. We've also had some discussions with OTC, the company that produces arbidol. and that what we'd like to do are to perform some detailed study of Arbidol against other viruses. So we have the HBV culture system is more or less up and running in the lab. We are working with Nick Crisp, who's got a liver slice culture of hepatitis B virus infected cultures going. We're thinking about doing some work on influenza in combination with a novel peptide inhibitors of hemagglutinin that David Baker's Institute of Protein Design has developed. And, like pizza, we, want to get into Zika. And what I really like to do is maybe use some next generation sequencing to profile Arbidol resistant mutations for HBV, HCV, and other viruses. So that's kind of an overview. There's other things going on in the lab, but I don't have time to tell you. But a lot of people have made this work possible. In particular, Jessica Wagner has driven much of this work over the years. We've had a lot of great collaborators locally, both at the UW and Fred Hutch, and nationally, UTMB, and really strong collaborations with these three natural product chemists at University of North Carolina at Greensboro. Some international collaborators and funding sources. And now this is the fun part of the talk. I said I'd mention a little bit about Canada and a little bit about hockey and music. So I was born in Canada. I came here. I've been in lab med since '94. Came here to do postdoc and never left. I'm actually a first generation Canadian. And I kind of inherited sport from my dad. He used to play semi-pro soccer. He was a goalie, and this is, in the late 50s, I think, in Brantford, Ontario. My mom was a musician. She played piano, and so I got kind of the guitar from her. So I'm a Canadian, first generation Canadian, now a US citizen since 2009, and being an immigrant, a child of immigrant parents, my dad thought, it's not soccer, I got to play hockey. So he pushed me toward hockey, and I still play hockey. I'm a active member in a local beer league. We play for the Miller Cup twice a year. It's made out of beer cans, and if you win the championship, you get your name engraved. My name is on here many times on a can of beer. It's very cool. So I'm keeping the sporting tradition going, and from my mom, I think I got the music side, so it's fun. My daughters are coming back to me now. They're kind of in their early to mid 20s, so they're starting to come to open mic with me, realizing that their dad isn't such a weirdo. And I also play. And you'll notice that this is the first time. This is actually at a scientific meeting, the International HCV Symposium. And if you look really closely, who's that right in the front row there? That would be Dr. Cookson. He got. So it's been really fun to do these things outside of science. So thank you for your time and attention. Appreciate it. Yes. Doing that right now, so we've also so that's a great question. In fact, some of the early Russian papers say that Arbidol is an interferon inducer, and in our original paper on Hep C, we looked at that. We looked at ISGs and HUH7 and some human hepatoma cells. Didn't see anything. Didn't see any interferon production. No effect on like ISRE reporter activity. That being said, we have some preliminary data from this LINCS. We've also had the LINCS people profile, that do the transcriptional profiling of Arbidol. And there's been in a few cell lines at the highest dose, and the highest dose tested was 10 micromolar, and that's within, that's non-cytotoxic. It's within the single digit to low double digit micromolar activity of Arbidol. That highest dose, some of the data look like when you ask it, what does your transcriptional signature look like most strongly? And it's come back on a few cell lines at only at this high dose that needs to be repeated, of interferon alpha, interferon beta, and interferon gamma. So, like NF-kappa B, say, other than like through IRF3 or something. Yeah, so it's making me relook at that now that we've got starting to get these links data. So those are in the process of being repeated. Yeah. Chihiro. Apparently, well, actually, in the PNAS paper that came out from Ian Wilson's lab, he mentioned a clinical trial that was registered at, clinicaltrials.org, but it was by a Russian. Like, I don't think there's anything being done here, and I'd like it to be. I've got really pure Breaking Bad, great stuff in the lab if you need any. Except it's white, it's not blue. Correct. Yeah. We focused on that for the bulk of the R01, and now we're starting to profile the individual components transcriptionally. I think that's, some people say, microarrays are just a fishing expedition, but if you really want to be unbiased, I could pick my favorite pathway and probably find an effect, but that's not going to get you at how they're really, at a fundamental level, how they're engaging cells. There are about 8 major components, 8. 7 flavonolignans and 1 flavonoid. Flavonoid is taxofolin, and there are there's psilibin A, psilibin B, iso A, iso B. There are some minor components in there that we may be studying if we can get them from another collaborator, but it's 8. That's the major component of the extract. There's also some lipids and stuff, but we're interested in the polyphenol type compounds. The natural product chemists that I work with do, and they get super pure mass spec and NMR validated compounds. Well, thank you all.